I'm here to show you how to do the blood type crossing and also the RH factor crossing. The first video will show you how to do the blood type crossing, and then the second video will show you how to do the RH factor crossing. If you turn to your lab menu to page 4, which you cannot see from the video here, at the bottom of the page 4, you will see the exercise that I asked you to do. Previous students use the separate paper to complete the exercise. After that, they just staple or attach to the lab menu when they turn in the lab menu by the end of the semester. So you can do the same thing. Otherwise, you don't have enough space to do all the Punnett square. So let's take a look at the blood type crossing first. I'll show you one of the example. Type A blood crosses with type B blood. A crosses B. So for type A blood, it has two cases. Okay, remember we went over it during the lecture, saying that type A blood is a dominant trait, and the type B blood is also dominant trait, and only type O blood is the recessive trait. So A is a dominant, so it can be homozygous dominant, meaning that it can be IA. We call this homozygous dominant. Or it can be IA and then small I. We call this heterozygous dominant. Homozygous because both allele, they are a dominant allele. Heterozygous because one allele is dominant, another allele is recessive. So for blood type B, it follows the same logic. Blood type B can also be homozygous dominant. It can also be heterozygous recessive, like this. Not heterozygous, heterozygous dominant. It can be heterozygous dominant, like this. When we ask you to cross A and B, it will generate 4 different cases, or 4 different Punnett square. Why? Because blood type A can be homozygous dominant, IA. When we cross with B, it can cross with IB, heterozygous dominant. This is case number 1. Case number 2 is homozygous dominant crosses with heterozygous dominant, IB small I. Third case will be heterozygous dominant crosses with homozygous dominant, IB. The fourth case, heterozygous dominant, IA small I crosses with heterozygous dominant, IB small I. So you need to show me 4 Punnett square in this case. So let's do all 4 cases together. IA, homozygous dominant crosses with homozygous dominant, IB. Here, homozygous dominant crosses with heterozygous dominant, IB, small I. Here, heterozygous dominant, IA, small I, crosses with homozygous dominant, IB, IB. And then heterozygous dominant, IA, small I, crosses with heterozygous dominant, IB, small I. Now, how do we fill Punnett square? It's very simple. We just copy everything. What do I mean by that? So IA is here, so we copy IA here. IB is here, we copy IB here. IA, IB. IA, IB. IA, IB. Similar in here, we copy the same thing. IA, IB. IA, IB. IA, I. IA, I. Similarly in here, IA, IB. In here, the tradition is that we put the dominant allele first. So we put the IB first, and then the recessive allele, small I, follows. IB. Similar, IB first and then small I. Here, IB. IB first. IA first. And then two small I. And then your lab manual asks you say the possible blood types the children could be. So let's take a look. In the first case, all four cases, they are IAIB. So 100% of the children will be blood type AB, which is the universal receiver or universal recipient, if you remember the lecture. Now let's take a look. 2 out of 4 cases, they are IAIB. Therefore, 50% of the children will be AB, and then 2 out of 4 will be IA and small I. That means 2 out of 4 will be heterozygous dominant of the type A blood. Therefore, 50% will be A. Now let's take a look here. 2 out of 4 cases will be IAIB. So it will be 50% of the children will be AB blood. And then 2 out of 4 cases will be IB small I. That is the heterozygous dominant for type B blood. Therefore, 50% will be B. In here, 1 out of 4 children will be IAIB. So 25% will be AB. And then 1 out of 4 will be heterozygous dominant of type B blood. Therefore, 25% will be B. 1 out of 4 will be IA small I. Therefore, 25% will be heterozygous dominant A. Last but not least, 25% will be small I, which means if you remember the lecture, recessive allele, if you have a homozygous recessive gene, that will be a blood type O. So that means 25% will be blood type O. So this is how you should do your exercise. In the next video, we will talk about RH factor, how we do the RH factor crossing. For the blood type, in the lecture I mentioned that this is an exception case for the Mendelian genetics because blood type is called co-dominant because both A and B, they are dominant allele, and then only O blood type is a recessive one. So it has two dominant. And blood type AB is the co-dominant trait. Yeah, if you have any question, feel free to send me a message on Canvas. I will test you about this on the lab quiz on Tuesday after the spring break.